we have Laurel Lietzenmayer, who will be, in her talk is resource allocation and development of dimorphic male sexually selected traits in the jumping spider, Maevia inclement. And I apologize if I get your name wrong or your species name wrong. I am very new to species names. Okay, take it away, Laura.
All right. So, hi, I'm Laurel. I'm a PhD candidate at the University of Florida with Dr. Lisa Taylor, and I study coloration and sexual selection in jumping spiders. So today I'm talking about jumping spiders, and these are a great system to work with in sexual selection because they have these bright colorations and patterns in males, and they also have these really complex courtship displays. And within sexual selection, we like to ask questions about these traits and how they might lead to mating success or mate rejection or even sexual cannibalism. And asking questions about these things can fall into categories of content and signals or hypotheses. And this list of hypotheses is really long and complicated, and they're not all mutually exclusive. So a couple examples could be, does this specific trait indicate current or past condition? Does it maybe signal species recognition? Or they could be efficacy-based, things like, is it a backup signal? Is it working as sensory bias or sensory overload? There's lots of different questions you can ask, but it's always a good place to start when asking questions about your specific species and specific morphological or behavioral traits. And an added complexity to that is if you're studying something that has dimorphic males. So you have two completely different strategies potentially, and species that are the males that look completely different and could maybe even be considered different species if people were to just look at them and knew nothing about them. So I just want to give a quick breakdown of Maevia inclemens and these two genetically determined morphs and what we know about them so far, because they've been heavily studied for a while now. So the tufted morph, as you can see here, courts typically further away from the female, stilts up tall and waves his abdomen underneath its body. And it's been shown that larger tufted males in better body condition are most successful, whereas overly eager males that have court quickly and more frequently have reduced mating success. And in field collected spiders, we've also found that bigger males have bigger tufts. There's 3 little tufts on the above their eyes there. And all of this information really well fits the literature and the model for an honest signal. The male is displaying quality of some kind to a female. But when you think about the striped morph and what we know about them, it gets a little bit more counterintuitive and complicated. So as you can see here, the striped male will start courtship from relatively closer to the female. It crouches and moves in a zigzag motion, and it's been found smaller striped males in poor condition are most successful, and they can increase their mating success by courting faster and courting more frequently. We've also found that stripes in a prey color choice experiment attract attention without increasing attack. So all this information has led us to develop 2 alternative reproductive hypotheses. The first being that the tufted male uses tufts to honestly signal their quality as a mate to females during courtship, whereas the striped male may use this coloration, black and white stripes on their legs, these long wavelength yellow-orange coloration on their pedipalps to reduce aggression or predation from females during courtship. And we might think this because these patterns are really common in aposematic insects, stripes and long wavelength coloration. So it might have a similar function in striped males without being actually toxic. But to even start asking questions about this behaviorally, we want to know what potential information content is found in these signals, and does that information align with these hypotheses? And that's where we're going to start. So what we did is a diet manipulation experiment to ask, how does nutrition during development affect attributes of sexually selected signals in each morph? And are these signals condition dependent? So with our diet manipulation experiment, we collected gravid females. The spiderlings emerged and dispersed, and they were fed pinhead crickets for 3 molts until they were large enough to be randomly placed into either a high-quality diet, which was a diet of fruit flies and pinhead crickets, alternating daily between flies and crickets 6 times a week. And those prey items were fed a diet of high calcium, protein, and vitamins. And then some spiders were put into a low-quality diet, which was only fruit flies that were fed a basic fruit fly medium 3 times a week. So this is manipulating quality and quantity. And the spiders were left on these diets all the way until they matured. And I just want to shout out to undergraduate researchers who fed spiders for hours and hours, for weeks and weeks, to make this possible. So if we want to think about the traits that we measured after they became adults, number one, we wanted to know, did our diet manipulation work? So we measured body size using carapace width as a body size proxy. This would tell us whether or not our diet manipulation was successful and ecologically relevant. So you would expect all spiders that were fed a high quality diet were relatively larger than ones fed a low quality diet. For the tufted male traits that we measured absolute tuft length, which is just the average length of all 3 tufts, and relative tuft length, which takes into account body size. And the striped male traits that we measured was achromatic contrast of the black and white stripes of the first leg pair, and the color metrics of the yellow-orange colored pedipalps, hue, saturation, and lightness. And I don't have time to get into it, but if anyone is interested in using Mica Toolbox or has used it before and wants to chat about it, let me know. It's a plugin for image data to measure color. So number one, we want to think about, did our diet manipulation work? And there's 2 things that we found that make us confident that that happened. We found that spiders fed a high quality diet were on average larger than the ones fed a low quality diet. And that is broken up between females, striped, and tufted males. And that was true all the way across the board. Another interesting thing is mismolting occurrences, which is something we didn't necessarily expect, but can occur. We found mismolting only occurred usually in the low quality treatment. And a mismolt could be a male missing a palp, as you can see here in the corner, or missing both palps, maybe missing a leg, or got completely stuck mid-molt and was not able to molt to maturity. And the fact that we're only seeing that in the low quality diet is kind of interesting. And it's something that we could discuss. So focusing now on just the tufted males and the tuft lengths, relative tuft lengths. So we have our high quality diet, low quality diet for both relative and absolute tuft lengths. We found high quality diet spiders had longer tufts and relatively longer tufts compared to their body. Which fits with what we've been thinking this whole time, that this is an honest signal, it's a condition-dependent signal. When we think about the striped morph, it gets a little bit more complicated. So thinking about achromatic contrast, black and white stripes were actually more contrasting in males fed a low-quality diet compared to a high-quality diet. And for our hue measurements, just a quick rundown so everyone understands this figure. Lower values in hue are correlated with a more red color over here if we think about LAB color space. So this angle here is hue. So if you have a higher angle, it's more of a yellow shade. A lower angle, it's more of a red shade. So red to yellow, not directly, but you get the pattern. We found that striped males fed a high quality diet had yellower pedipalps compared to the low quality diet striped males, which were more of a orangish color. And thinking about saturation and lightness, we found striped males fed a high quality diet had less saturated pedipalps, And in lightness, there was no difference here. We also wanted to take some control measurements from a non-signaling trait. And this was just a patch on the back of the femur of the front leg. And taking the same exact measurements here, we didn't see any difference between lightness, contrast, hue, or sound values. So this indicates that there's something interesting and important going on with those sexually selected traits, not the non-signaling traits. So back to this figure here, did our diet manipulation work? Yes, we're pretty confident it did. There was body size predictions that matched what we would have thought, and mismolts occurring predominantly in that low quality treatment versus the high quality treatment. As for the content of the puffs, we found that this closely fits an honest signal hypothesis, as all the other information on tufted males does. Striped morphs, still kind of a mystery. Maybe there's trade-offs going on with body size. Maybe it's linked to condition, but the patterns are just a little bit more complexing. And this still doesn't totally falsify the idea that this is an aggression deterrent hypothesis in terms of their morphological traits. So some other tidbits that I just want to throw out there, if anyone's interested, the molting issues, it might have affected some of our results because it affects the CD integrity of low quality males. Maybe the tufts are breaking off or the CD on the palps is breaking off during the molting process. And that's what led to the pattern. We also wanted to break down between the black and white stripes, which one was driving the patterns of contrast more. So we found that the white stripes were actually more affected by diet than the black stripes were. Which is kind of interesting to think about and could lead to more specific questions that you could ask. And the one big obvious question is, do females even pay attention or care about these traits to begin with? So if anyone's interested in that question and the behavioral aspects of these hypotheses that we put forward and what the information in these signals really means in a courtship context, come to ABS in August and I will show you that. And I just want to shout out my lab, undergraduates who made this possible, and the ISA Oscar and Frank Student Research Fund, and obviously the spiders that participated because I got pretty attached to them from raising them from babies all the way to adults. And
great, thank you, Laurel. That was really interesting and perfectly timed. So the first question we have is from Sue Kagani, and she says, can you clarify the low quality diet? Was it just flies?
Yeah, the low quality diet was just fruit flies, 3 fruit flies 3 times a week. And those flies were only fed a basic fruit fly medium.
After I have a follow-up to that. So the high quality diet was the fruit flies and the crickets, right? Mm-hmm. So do you know whether it was primarily the crickets that was contributing to the improved diet, or?
We really don't know. So that's something that you could break up and feed just crickets, just flies on those same diets. But they were alternated between flies and crickets the whole time. And it took them months to mature. So I'm piecing those apart is not possible with what I have, but it would be interesting to look at.
All right. We have another question. Why did the tufted morph not court closer since it's an honest signal?
So there's a couple things to think about. One is the field of view of the female. So the height at which they're courting could affect how close they want to be to the female. Another thing is cannibalism risks. So if they're courting and using an honest signal strategy, they want to make sure that the female's assessing them from a safe distance. Whereas the striped male is kind of a mystery. Maybe if it is a predator deterrent, that allows them to court closer. But at this point, we don't know. But the field of view is something that's been shown before.
Great. Another question is, Could it be the final size and male morph related to the numbers of molts in each group, if it is variable, or it was the same for both diet groups?
It was pretty much the same across the board. All of them molted with 6 or 7 instars, and there wasn't really a huge difference between the low or high quality diets.
And the question is, what determines which morph a male will develop into?
So it's genetic. So there's a bias towards the paternal morph. It's typically like I want to say like a 30-70, 60-40. Don't quote me on that, but it's genetic. So there's a bias towards whatever the paternal morph was. You can still get both morphs from the same egg sac, but it's a chromosomal thing.
And I missed this. As a heads up, the track 2 talks are running about 15 minutes behind. So keep this in mind if you're planning on switching rooms for a specific talk. All right, I think we have enough time for just one more question here. Could stripes in one morph be mimicking tufts in another? Or could the female be misreading stripes as tufts?
Huh. That's an interesting question. I never really thought about that before. But yeah, I would have to think about that more. Yeah, I don't know.
Okay, great. Well, thank you so much, Laurel. I really enjoyed that. The